We're going to do that. All right. Hello, everybody. We're back in our neuroanatomy course here. This is going to be lecture 2. Before we do that, I just want to congratulate Bhagyashree, who just got accepted to Hopkins for fellowship in neurocritical care. So good for her. And also, we have a little bit of business. Same person, Bhagyashree, has reminded me that there are clinical correlations as part of this course that are taught by all of you, the residents in the program. And so when we get to regional anatomy, medulla, you will have a correlation on strokes of the medulla, strokes of the pons, things like that. Today, however, we're going to talk about glia, and let's get started straight away. So, right, as in started straight away. We have a pretty, can I get rid of this thing?
Yeah.
Hit that,
Right.
Or no?
Yeah,
Hit X. Good. A pretty extensive outline, but the usual length. We're going to talk a little bit about the history and some of the concepts related to glia. We will then talk about cell lineages in general, neuroepithelial cell lineages, and glial physiology. We're going to look then at a lot of structure and the function of astrocytes and glia, radial glia, the other glia that play a role in development. We're going to look at oligos, and then very briefly micro, and I made that small because microglia are very small. And I guess I'm working with this. A lot of information on this page, but that's okay. Very simple. The central nervous system essentially consists of 2 cell types. We spoke about neurons last week. We'll talk about glia today. The term glia, sort of as you can hear, it really initially was glue. That was the idea. These were first described by the great German pathologist Virchow. He didn't know what they were. He thought that they were connective tissue of the brain. There is no connective tissue in the brain, in the central nervous system. There is none. But he was certainly on the right track in terms of supportive structure, and we'll define that a lot more. Eventually, it was recognized that the glial cells separate neurons from the vasculature. Neurons do not come in direct contact with the vasculature in any setting that I can think of. There's always a glial cell interposed between them. Then it was realized that glia outnumbered neurons by quite a bit. In all regions of the adult human central nervous system, there are about 10 glia for every neuron. And although it's 10 to 1 because they're smaller than the neurons, they make up about 50% of the brain volume. The variation is considerable, and it is, I think, coincidental that the extremes are in the putamen, where you have 4 glia for every neuron, and the globus pallidus, where you have 100 glia for every neuron. And yet, isn't this strange that 2 structures that are next to one another have the highest and lowest ratios? But this brings up an important neuroanatomic point. Last week, I made the point that the language of neuroanatomy is subtle. The putamen and the globus pallidus are derived from different embryologic structures. In the adult, that's not obvious, but in the embryology, the putamen is derived from the telencephalon. The globus pallidus is diencephalic. So although they're in the adult, they're next to one another, their embryology is quite different. And that is going to be a recurring theme. We're going to be talking about that throughout the course. Glia increase in number through phylogeny. So this is another principle. You might think that as time goes by, for instance, you might see a picture of a shark, and you see in the picture of the shark this great big diencephalon and this little bitty telencephalon overlying it. And then in the human, of course, the telencephalon is so huge compared to the diencephalon. But that might give you the false impression that the diencephalon has become smaller as we advance in phylogeny, and that's not true. Only the relative size decreases, but the actual size increases enormously, right, as we get closer to the human in phylogeny, all right? So that's going to be a principle that you see. And so you can think of glia as a more primitive anything, because glia actually increase in numbers as we go through phylogeny. The greater the specialization of the neuron, the greater its need for isolation from the rest of the world, the more glia have to work in order to support it. And that's the theme of today's talk, is that the glia are supporting the neurons. The glia also appear first in ontogeny, that is in the development of the fetus. But then the majority of glial cells will appear after all of the neurons have arisen. And we will also periodically use this principle that ontogeny recapitulates phylogeny. You see the phylogeny occur in the development of the embryo. This is very strange. Okay, let's talk about glial types. So macroglia are the real glia as opposed to microglia, right? Macroglia then they all share a neuroepithelial origin with neurons. The astrocyte, the oligo, the radioglia, and the ependyma, we're going to talk about all of them. They are all neuroepithelial cells. The microglia is the only mesodermal tissue, although outside the brain, the dura is going to have to be separated from the neuroepithelium, and we're going to look at that interface very shortly. All right, let's look at some pictures. So we see a couple of astrocytes here, two different types, the fibrous astrocyte and the protoplasmic astrocyte. There's the oligo and there's the microglia. Now, morphology is not a good way of identifying glial cells. They change their shape and their phenotype so dramatically and so quickly that it isn't a good idea. So if you want to know that you're looking at a glial cell, you really need to stain the cell. For astrocytes, we use GFAP. For oligos, you have many other choices to use. All right, so you use these biochemical stains, whether you're looking at light or EM. For EM, the nuclei look a little bit different, but there's nothing really reliable. I'm going to show you pictures of what the standard classic is, but they are quite variable. And so staining is important for glia. It is the case, so Virchow was certainly onto something, and it is the case that glial cells isolate the neurons so the neurons can perform their highly specialized function of information transfer. But the neuron, in that extreme specialization, has virtually lost the ability to take care of itself, right? And all of that work will be done by the glial cells. Those are the glial cells physically isolate neurons from the world around them. They provide support. They provide protection against various kinds of insults that may try to work their way toward the neuron. The glial cells maintain the metabolic and ionic environment in which the neuron can function. And the neuron is not capable of maintaining its own homeostasis. It's simply not capable of doing that, and is completely dependent on the astrocyte or oligos for this. The glial cells also contribute directly to neurotransmission. One obvious one is the myelinization by oligos, but another equally obvious one is the role of the astrocyte in terminating the synaptic signal by removing neurotransmitters from the synaptic cleft. So the glial cells are not only there at the behest of the neurons to protect them, but they do get involved in neurotransmission. Glial cells also were responsible for guiding the central nervous system neurons to their locations during development, and they participate actively or sometimes in a very negative way in regeneration and plasticity. That is, the wrong response of a glial cell may inhibit regeneration, and that may have to be overcome. The neuron in that case may be willing to regenerate, but unable to because of obstruction by glial cells. Eventually, during the course, we'll take up all of these topics. Let's focus on astrocytes. They are the largest and by far the most numerous of the glial elements. They have a stellate shape, hence the term astrocyte, okay? And you'll see that. I'll show you pictures and you'll go, oh yeah, that's Starry Night kind of stuff. There are two varieties that we've already seen briefly, that is the fibrous and the protoplasmic astrocyte. Those are more about shape. They're a little bit about function, but again, the glial cell, the astrocyte in particular, is so polymorphic that you really can't say much about it based on morphology. it expresses the GFAP, and that's how we're going to recognize it. Another way to identify astrocytes, however, is that astrocytes have gap junctions, and with those gap junctions, they form a electrochemical syncytium, a real syncytium, so that they communicate with one another, can influence both the resting membrane potential and also the calcium concentration of the cytoplasm. And they share that, and it's a syncytium. Once it occurs in one cell, those other astrocytes are going to directly communicate with its neighbors via gap junctions and things like that. Okay, so they are a unit. Once they don't stay localized. Once one of the cells feels this, and you can see it in a lot of cell culture situations, you can prick an astrocyte, and all the astrocytes respond almost instantaneously. You can see the wave go away from it. It's quite a dramatic effect. They have typical organelles. They have the astrocyte has a finely granular nucleus. There are nuclear pores, there's mitochondria, RER, smooth ER, glial filaments, very few microtubules, and they will have granules in them. Right? GFAP stain of astrocytes in tissue. And you could see why they're called astrocytes. They're completely stellate. I mean, that's just the first impression that you get. Here's our friend the astrocyte in a EM. And first of all, there's a fairly scant cytoplasm in the cell body, right? I mean, here's the nucleus. Here's the outline of the cell. The rest of that, all that out here is dendrite. There's some cytoplasm in here. But other than that, it's all nucleus and a little bit. Okay, that's not uncommon. And here's an oligo, by the way. And this is a pretty conventional. The oligo has a fairly dense cytoplasm surrounding this nucleus. This is a fairly typical. I'll show you more pictures, but we'll get there. What's the difference between a fibrous and a protoplasmic astrocyte? And it really has to do with shape, as I say. There are differences. So the processes of the fibrous astrocyte are fewer and longer. That's what makes it a fibrous astrocyte. In general, the processes are fewer and longer. They have less branching. Not that they don't have a lot of branching, they do. But compared to the protoplasmic astrocyte, you'll see they have much fewer branching and therefore longer processes, really, is what they are. And they can radiate for quite a distance. With that, they're going to have neurofilaments, and yet I've already told you that they're very pleomorphic. They change shape very quickly. Last week, we talked about neurofilaments as being a more stable part of the system. Microtubules are faster. Why doesn't it use microtubules? Well, we didn't get into it, but neurofilaments have ways of stabilizing themselves or not. And so in the astrocyte, they are not stabilized. They're not cross-linked as they are in a long axon, for instance. And so there are differences. In the fibrous astrocyte, if they have a nucleolus at all, it's generally small, and they have these small granular swellings at the light level, which are known as gliosomes, okay? And basically they're clumps of mitochondria. And then these are the cells that are interposed between the neuron and the adjacent capillary. They have very prominent perivascular end feet, and we're going to see this. We're going to look at this in more detail. The fibrous astrocyte also is more numerous in white matter. So here you go. These are fibrous astrocytes. You can make out pretty easily these processes. They branch. You know, they do branch. And you go, I could say, look at this and say, wow, if this is fibrous, what the heck does a protoplasmic astrocyte? This looks, we'll get there. Here's a beautiful picture. Again, a GFAP stain of just a astrocyte caught in the plane of the slice of the section of the light level. Very pretty. There's another one, silver stain, allowing you to see the whole astrocyte. And again, astrocyte. And here's a multiple fluorescent labeling stains of an astrocyte. It's a fibrous astrocyte, and a fairly small number of processes heading away from the cell body. Here's EM, again, here's your astrocyte, fairly scant, eccentrically placed nucleus, so you have scant cytoplasm over here, but then you have this, here's the beginning of a process taking off, And you can see all of the normal machinery. This would be a gliosome. It's a clump of mitochondria, rough ER, a smooth ER, all the things, glycogen granules, that sort of thing. It's all there. All right, a couple of lysosomes. All right, darkly stained, sorry. The protoplasmic astrocyte, which also can be called a mossy cell, has numerous freely branching processes in comparison to the fibrous. It also has a perivascular end feet and is closer to the cell body or the proximal dendrites. So you're looking at the fibrous astrocytes are going to be mainly in white matter, so supporting axons and oligos, and the protoplasmic astrocytes closer into the gray matter, right? Because they're near the cell bodies and the proximal dendrites. See how different they are? And here we are. We're up in cerebral cortex. Here's a pyramidal neuron, apical dendrite, basal dendrite, right? We don't really see the axon. Maybe that's the axon right there. But it's surrounded by these protoplasmic astrocytes. And these branch. These are way different than the fibers. And they will have the perivascular end feet separating the cell body and the proximal dendrites from the vasculature. And it's just a picture. Is this a protoplasmic astrocyte? I have no idea. It's a picture of an astrocyte, small cell body, okay? There's another astrocyte, more cytoplasm in this case, but there's really nothing distinguishing. I mean, there really isn't anything distinguishing. Here's some neurofilaments running through the astrocyte, through a process, an astrocytic process. Yeah, that's a good illustration of the neurofilaments. Okay, now I want to, staying with the theme of the astrocyte, we're going to move to the macroscopic view here. And we're going to look at the inner and outer surface of the brain, right? So now we're not just talking neuron or glial cell. Now we're talking about the macroscopic surfaces of the brain. In other words, the pia mater and the ventricle. And we want to look at how those are formed with the help of glial cells. So they isolate the central nervous system from the outside world. The pia mater is the innermost layer of meninges. Therefore, it is mesenchymal, right? This is not the pia mater, those cells, they are a single interrupted layer of elongated cells, and they lie flat and close to the surface of the cortex. But they are mesenchymal, and they're going to have to be separated from the neuroepithelium. They are separated by a basal membrane, and the basal membranes are really interesting. Basal membranes are nearly acellular structures, a lot of collagen, type 4 collagen. They're really interesting. I was reading a lot this week on the basal membrane, and the basal membrane is formed by both the mesenchymal tissue and the neuroepithelial tissue, both of which are touching it, and they both contribute. And so the basal membrane, on one half of it, is mesenchymal, but on the other half of it, the other face of it, is neuroepithelial and meet in the basement membrane. Which is very cool. I think it's very cool. So that's the meeting. East meets west in the basal membrane, not basement membrane, but basal membrane. All right. So we have the pia coming from the meninges side, coming from the brain side, the parenchyma side, is going to be the glia limitans. And the glia limitans is formed by fibrous astrocytes, okay? Fibrous astrocytes, right? These are the marginal glia, and they're going to send a very short, stout process toward the pial surface, and when they contact the basement membrane, not the pia, but the basal membrane, then they're going to give off these huge end feet, which will firmly attach to the basal membrane via a series of junctional complexes, including, and then between them, they're going to have desmosomes. Remember, astrocytes can form a syncytium. So if they want to get tight and adjacent to one another, they can do that. And this is a really good picture. We have several really nice pictures to work with. So stay with me. Here's the pia. All right, this very light outer surface is the pia. See, we're on the surface of the brain, right? This is the pia. This is the basal membrane. These are the glia limitans, the foot processes of the fibrous astrocytes. Okay, and then they, of course, will also contact the vasculature and get into, and we'll look at the neurovascular unit in a minute, all right, in a very short minute. Right, but you see this structure? So again, pia, basal membrane, glia limitans. Just a bunch of pictures to show you. This is a histology. Here's brain. All right. Here's the glia limitans. Here are their foot processes. You can almost, we're at light level. You can also almost make out the basal membrane. And then this is the pia. All right. And here you go. This was better. This is, I thought, a better tissue. Here's pia. Here's the basal membrane. Here's the glia limitans. Very pretty. Very nice. Okay. There's a cartoon. I don't think the cartoon's as good, but it sort of gives you the sense. I kept it in here for this reason. Here's the basal membrane. Here's the glia limitans. Here you are at EM, and you're dealing with pia. This is and this is basal membrane, and this is glia limitans. Okay. Very nice. Basal membrane. Yeah. Okay. Brain. All right. Now you got that as the outer surface of the brain. Outside of that, just think what's outside the pia is the arachnoid. That's where the blood vessels are. The brain is going to need that blood flow. So somehow these vessels in the arachnoid space are going to have to get down into the parenchyma to make a capillary. But they can't be in touch with the neurons. So how do we do that? That becomes the Virchow-Robin space. And now, so what you want to think of is that these blood vessels in the arachnoid state are going to insinuate themselves down into the parenchyma. But when they do, they're going to pull the pia, the basal membrane, and the glia limitans with them. All right? That's what we're going to see. That's the Virchow-Robin space. So it is basically all the arteries and veins penetrating the brain from the subarachnoid space, and this invagination, that's the word I was looking for. It's an invagination of that whole three-layer structure down into the parenchyma, okay? It has to be separated, all right, we've been through this. And the Virchow-Robin space, and you can see these spaces, it's real space, is what makes up what's called the glymphatic system of the brain, which is very important. There's a whole theory right now that there's impaired glymphatics in Alzheimer's disease, and toxins are not removed, and and there's pretty good evidence for this sort of thing. And I remember one of my very close friends was one of the people who conceived of this idea when she was a fellow at Cornell. And I've just followed her and her career with this idea. She's stayed with us forever and really has made major contributions in doing it. All right, here we have a pia artery. All right, so here's, notice this is the blue. This is incomplete. So what are we calling the blue? The blue is the pia mater. So it's barely showing you this white in between will be the basal membrane. And here's your glia limitans, right? Okay, here's a penetrating artery. So first of all, it's not in direct touch, which brings the subarachnoid space with it, right? And so here's the pia. It's not directly in touch with the pia. This space right there, that's the Virchow-Robin space. All right, eventually, that space is going to become obliterated and the pia will be in direct contact with the vessel, all right? And that's not useful, right? Because the pia is still isolating the vessel from the brain, from the parenchyma, all right? So eventually the pia is going to have to disappear too, and the basal membrane won't disappear. The basal membrane is going to stay, all right? But most importantly, the astrocyte is going to become in not absolutely direct, but in pretty damn close to direct contact with the capillary. And of course, the capillary, as you know, when we talk about blood-brain barrier in a minute, the capillary is tight in the brain. It's not just loose and letting fluid and serum run out into the space. Not at all. It's tight. All the endothelial cells have tight junctions. So anything now coming from this space to this space is going to have to be transported by specific transporters. And that is the blood-brain barrier. Okay, so you're now looking at the anatomic basis of the blood-brain barrier. Here we have a artery, see, muscle, right? All right. So this is definitely still an arteriole working its way toward the parenchyma of the brain, but not in contact. Here's the pia. right? This is the pia here, all right? This is clearly an arteriole, all right? And this is the Virchow-Robin space. Okay, that's the space. So it brings the arachnoid space with it into the brain substance, and then until it eventually contacts the pia, no more Virchow-Robin space. The pia then goes away. You have a basement membrane, and the glial cell, the astrocyte, are now able to form a direct relationship with the capillary, with the blood vessel as a capillary. That's how it works. All right. So let's just focus on that for a minute. And we're going to call that interaction of the astrocyte with the capillary the neurovascular unit. So you have the neuron, you have the capillary, and in between is the astrocyte. And the astrocyte is in intimate contact with the capillary through a series of specific transporters and channels and things like that. And then it's going to do different transporters, different channels, okay, but all specific in contact with the neuron. And that's it. So the astrocyte is sitting there, sucking things out of the capillary, giving it to the neuron, taking things back from the neuron, and putting it in the capillary. That is the neurovascular unit. That is the basis of all brain activity. Okay, that's it. This is where this is. My father would say in Yiddish, we get to tachas. This is where it happens. Okay, all right. That's all I can say. That's the idea here. You have this additional cell, the pericyte. The pericyte is not a neuroepithelial cell. It's found throughout the body. It is a supporting cell. Really is almost, not exactly, but almost. This is basement membrane, not basal membrane. And it provides a basement membrane to the capillary, and it remains. It's still there. See, even though the astrocyte wants to be in direct contact with the capillary endothelium, it isn't quite. This orange here is basement membrane, which the pericyte has generated, and the astrocyte is. So obviously this has to allow transport of things through here. An exact role I have not been able to find yet. It seems to be, first of all, the pericyte is contractile, so it can open or close the capillary to some extent, increase it or decrease it. It participates in immune responsiveness for the tissue, right? It's also partly under control of the brain, so it's an interesting interaction, and I have never found a lot of good data on this. It's a tough thing to study because that's something you'd have a hard time studying in vitro. Astrocytes contacting capillaries. Lovely. Do you love it? Look at how pretty. Look at that. Isn't that gorgeous? So you can see how the astrocyte foot processes invest the capillary. That's beautiful. And astrocytes, do you want to call them anything other than astrocytes? No, they're just so stellar. And so it's really pretty. This is beautiful anatomy. That's gorgeous. Here we are at the EM level, and all the elements are here. We're in the lumen. We can see the endothelial cell. We can see the basement membrane. We can see the pericyte. We can see the astrocyte. All right, astrocyte, all of that. Okay, so this is a great picture. Tight junctions of the astrocytic processes. Could be from the same astrocyte, could be from a different astrocyte. Okay, got it? That's the anatomy. That's it. Here we have another one a little bit higher. Endothelial cell, basement membrane. Look at here. Look at this. Beautiful view of the basement membrane. Glial process, astrocytic process here. All right. This is probably pericyte here. Collagen tissue. Collagen. This is all collagen. All right. Great. All right. That's at the outer surface. Now we want to go to the inner surface. It's even more complicated. It's really kind of cool, geometrical. So now we're looking at the ventricular surface of the brain. Again, another astrocytic function. We start with a simple layer of cuboidal epithelium lining the ventricular surface. And we say epithelium, and I didn't say neuroepithelium, but epithelium is epithelium. It's not mesenchymal. So we're okay, apparently, but we're not going to actually have the epithelium in contact with the brain per se. and yet we're going to make CSF and absorb CSF through this thing. So this is going to be interesting. So let's see what goes on. The apical surfaces of the cuboidal epithelium of the cells are covered with both cilia and microvilli. The cilia are going to help circulate the CSF, and the microvilli will absorb and secrete CSF. Microvilli, so cilia are long structures. They are about 10 microns long. Microvilli are only about 1 micron long. So you're not going to have trouble separating these two. When we get good histology, we're going to see them as very different. The ependymal cells, okay, are going to make a brain CSF interface. We're going to have specialized lateral junctions for the epithelium. All right. We're going to intertwine the astrocytes in the subependymal zone, but it's going to remain a subependymal zone. We will not talk about astrocytes, with the exception of the tanocyte. We are not going to talk about astrocytes meeting the ventricular surface, the real surface. They're not going to be in contact with CSF. There will always be an epithelium in between. And so we will always be saying that the astrocyte runs from the subependymal surface to the pial surface. It does not contact CSF. All right, here's the epithelium. Simple stuff. You could almost say, gosh, could that be in the gut? And then, well, yeah, I mean, actually, in a certain sense, at the light level, it could. There's a single cuboidal layer of cilia, and you can't make them out individually, but you get that this is fuzzy. That they're fuzzy because those are the microvilli. Cilia are not fuzzy, they're cilia. All right. Here, just to give you a sense of some of the tight junctions that exist between the epithelial cells, the zona occludens, okay, and the zona, and there's a zona obliterans. One of the really interesting things is that, and I haven't found a good picture, but in some of the cells at in the ependyma, the cytoplasm has been extruded from the cell, and one cell membrane is adherent to another cell membrane of the same cell. In other words, what I'm saying is there could be a cell in here, but the cytoplasm has been removed. The zona obliterans removes all of the cytoplasm from between the two surfaces of the cell. Very peculiar arrangement. And then you have all vesicles for transport because nothing's getting in or out. And here's a scanning electron microscope with gold toning, the kind we previously talked about. And you can see the cilia very easily. And if you look underneath the cilia down here on the surface are the microvilli. I think I have a better picture. Yeah, I just found this yesterday. This is a really gorgeous. I mean, wow, it's like I'm almost throwing the other ones out. This is it, okay. Microvilli, I put the bloodlines in and cilia. That's pretty. That's beautiful. Again, some good anatomy in this. This is just to show you the arrangement here at the ependyma and the subependyma. Okay, these astrocytes are not going to make it to the surface. You can see the zones, the occludens. Okay. Sitting down here, vasculature is outside. Now, in a minute, we're going to struggle with the concept of choroid plexus. Okay, so we're going right to that. We're going to keep going. And this vessel is going to have to participate in the choroid plexus. And so we're going to look at that in a minute. Now, how does this happen? All right. Remember, you have these blood vessels, which have now made their way from the arachnoid space. They had basement membrane. If they haven't gotten to the capillary level, they still may have basal membrane. They still may have the, glia limitan, all right? And now they're going to push their way all the way through the entire brain parenchyma until they contact the epithelium of the epithelial lining, the ependyma, if you will, of the ventricle. And then what happens, okay? So this is known as the tela choroidea, all right? That is where the pial layer has migrated with the subarachnoid vessel, has migrated all the way through the parenchyma of the brain until it's in contact with the other surface, the inner surface of the brain, which is the ependymal lining. And as it does that, it's then going to, furthermore, it's going to invaginate. I actually brought a couple of gloves, but it's as if the artery is my hand, and the hand puts on a glove, and then it works its way through the muck, okay, which is the brain, in this case is the muck, until it reaches the other surface, and then it puts another glove on. It double gloves, and one of them is the pia. Okay, there's a blood vessel, and then there's the pia is my inner glove, and the ependymal lining is the outer glove. And there's nothing in between the two gloves, okay? But inside the pia is the blood vessel, okay? That was the analogy. I actually, like I said, I brought two gloves to do this with a white one and a purple one, and I thought it was, never mind. You get the idea, all right? So this is going to invaginate its way in, and then, and that's it. Then the choroid epithelium will be derived from the ependymal vessels. The blood vessels of the choroid will come, like all others, from the arachnoid space, carrying pia with it. And then they can do their own thing and set up a set of secretory proteins and absorbing proteins, and you can create the choroid plexus. And I found this picture yesterday. These are some new pictures which have really, really helped get these points across. And here, this is just it in view. Here's the brain parenchyma. Here are astrocytes, right? So this is the parenchyma. Notice, so here you're going from arachnoid, blood vessel, all the way into the CSF, okay? This is the ependymal lining of the CSF. This is the frond-like choroid plexus, right? And you have inside it a blood vessel, a pial structure, an ependymal structure, and nothing else. Isn't that great? This picture really, I replaced about 6 other pictures with this one picture because it gets you there, all the way there. I love this picture. This was one of the old ones that I had. You can just see this is arachnoid. This is mesenchymal tissue, all the way. The mesenchymal tissue never goes away. They're your blood vessels that it carried with it. They are on the arachnoid side of the mesenchyma, which is mainly pia, right? And then the pia has a basement, the basal membrane, and then that is in contact with the epithelium, the ependymal lining, and that's your choroid plexus. And these are just more, this was a new picture. Also, there's a space where you have all these things being transported and CSF being made and absorbed, and great pictures, zona occludens. So great pictures. Here's a real histology. Here you are with the ependymal lining of the brain where there is no choroid plexus, right? That's in the ventricle. This is ventricle. This is brain. Could be the head of the caudate, right? All right. So in contact with the CSF, not quite in contact. You have an ependymal lining between them. But here you have next to it the choroid plexus, and you can see similar types of cells. They're different. All right. You can see pia. Beautiful. And vessels, blood vessels. Okay. Choroid plexus. So really great pictures. And here you are, and you can have it right from the pial lining to the ependymal lining with nothing in between. That's your choroid plexus. All right. There's arachnoid space, if you will, in between. They're not arachnoid. Okay, on to the next topic, radial glia. Now, radial glia are complicated. So let's start with that. And radial glia are still being defined. This is if there's lots of research, plenty of research to be done on glial cells. If the most active area by far, in my mind, or the most complicated or complex is the radial glia. Now, the radial glia are neuroepithelial cells. They are, many of them, depending on what you mean by radial glia, they are developmentally, they're stem cells, is another way of, these are at one stage, these are the stem cells of the brain, all right? They are at one stage. They're neuroepithelial, they're already committed to neuroepithelial, but they are not committed to neuron or glia. And we're gonna have a whole lecture on this. We'll be doing plenty of embryology in this course, you need it. But at any rate, for now, the point is that they span from the subventricular zone to from the all the way from the subependyma to the subventricular zone. they're going to go from pia to the ependymal lining. All right. They are progenitor cells at some stages. Then they settle in. They can make neurons. They can make astrocytes. They can make oligos. They can generate more radioglia. At a prenatal stage, they will start to stain for GFAP. Now they're moving on. They also, radioglia will persist in the cerebellum as the Bergman glia, big structural glia. And they remain dormant in the adult nervous system, although they are thought to contribute a great deal to gliosis. So when you start generating astrocytes and that sort of thing, the radioglia will become active again. And during development, they are also the main guidance. Because they run from one surface of the brain to the other, the neuron can encounter them and go to whatever layer, if you will, of the developing nervous system, and then hit their target biochemistry, recognize they're in the right place, jump off the radioglia and say, I'm staying here, thanks for the ride. Here you go. So here's a radioglia. This is Pascal Rakish and his wife at Yale, generated most of the concept related to the radioglia in their first formative years. Here's stains from their research, and here's a cartoon of progenitor cells breaking off from the radioglia in the subependymal and the subventricular zone. That's why you in the neonate, in the premature infant, you have the periventricular hemorrhages. Okay, this is because this is what's going on in there, and it's a very soft structure, very not developed yet at all, not astrocytic. And so at any rate, and then here you have neurons migrating up to their place where they belong. Okay, we're almost done. Let's talk about the oligo. The oligo is, as you could imagine, has very few processes, and that's why it's called an oligodendrocyte. That's its real name, the oligodendrocyte. We call them oligoglio, that's okay. Oligodendroglia is also whatever you want. They're all okay. They'll respond equally to any name that you want. They are small, they are regular, they are pear-shaped cell bodies. They have fried egg appearance in certain settings. They are they have a scant and densely stained cytoplasm and lots of mitochondria. And so this is metabolic. They themselves are metabolically active. And I want to develop this concept of metabolism again. Where do they sit? Okay, are they like the neuron, just sort of helpless and a parasite of the rest of the astros? Or are they like the astrocyte? No, they're not like the astrocyte. They have a specialized function that is to make myelin, but they have a second function that is a biochemical function that we're going to come back to in a minute. All right, they're metabolically active. If there's a lot of mitochondria, then they're metabolically active. They have ribosomes, so they're synthesizing things all the time. Golgi, microtubules. All right, there are no neurofilaments. There is no glycogen. So if then they too must be dependent on the astrocyte. And yet, okay, we're going to come back to the and yet, which is a very interesting one that we're dealing with clinically today. So here you go. They make myelin. You know that. Now, a single oligo in the central nervous system can myelinate 70 to 70 axons. So again, unlike the Schwann cell, which can wrap itself around and migrate the cell body around and make a, you can't do that, right? It's topographically, it won't work. It's going to make a mess, right? So instead, it's going to, the myelination, I'll show you a picture, but the myelination is going to be an inside wrapping, not an outside wrapping. Processes that connect the cell body to the myelin are so small that they virtually disappear. And we're not going to see them. They're scant, all right? And the myelin will become apparent. We're going to look at developing myelin in a minute. But the second function of the oligo, which is neglected but clinically important, and why I'm so interested in this patient I'm taking care of in the ICU today, is that the oligo is responsible to regulate pH, not the astrocyte. The astrocyte is busy. The astrocyte is generating ATP. It's taking care of the intermediary metabolism of energy. But with that, it's going to be consuming glucose very often anaerobically, and generating acidosis. And it isn't in the process of generating the acidosis, it can't be the one who's going to solve the acidosis. Somebody else is going to have to solve the problem. And it's the oligo which takes on that responsibility. So now we see a symbiotic relationship between the astrocyte and the oligo to maintain the intermediary metabolic homeostasis of the brain. Very pretty, okay? Neuron only takes. In this regard, the neuron only isn't dealing with it. It's a, like, take care of me, guys, okay? That's the way it is. The oligo also, however, will express neurotransmitter receptors during development. Here's the oligo, and here we are obviously at the light level. Here's a neuron, now again, a large pyramidal neuron. You can see the cell body. And here's the oligo with a few processes surrounding it. This is probably in developing tissue, very likely. Here's the oligo and its processes. Here's the, I promise you, a more typical oligo, densely stained cytoplasm. All right, ribosomes all over the place. Okay, nucleus. This is an astrocyte. This is not the oligo. This is the astrocyte. All right. Here, a diagram of what you already know. And here's the myelin. Notice it's wrapping itself around. This is the leading edge. The inner edge of the myelin is just twirling around the axon because it can't do it with the outer edge. It would make no sense because it's connected to 70 different axons. The one wants it to go this way, so we can't do that in the central nervous system. Let's just look at some pretty pictures. Electron microscopy. Here's an oligo. Here's a relatively mature myelin. This is an astrocyte. Here's an immature myelin. You can actually see the leading edge of the myelin right here. Right? So it's doing its thing, investing the axon. Here's it's barely been around a couple times, cell body, migrating. So probably going that way. Here you go again. You see? So that's that migrating edge working its way in contact with the axon and constantly in contact with it. Here you have sort of a bulb, if you will, of oligo making investing multiple axons simultaneously, each of them therefore having to invest their, see the cell bodies going to get started. Relatively more mature cell body working with. So notice I'm not saying nucleus, okay, but cytoplasm here investing its way around the axon. Various stages. Here's just very, very early myelination, because these are all unmyelinated, and then you start to see a little myelination form. You can see that myelin takes up a lot of space. Okay, this is going to have to grow substantially for all of these guys to get myelinated. Is going to take up a lot of space, right? Going to really take up a lot of volume. Right, different stages. Again, that leading edge. Right. And then here's the mature myelin. I love this picture. You can see inner and outer, dark, dense and light line. You can still see where that inner edge is, right? Contacting itself and migrating over itself and just making more. But that's pretty mature myelin, compacted. That's what you say, compacted. That's what you call it, compacted. Here's a compacted node of Ranvier. I showed you this picture last week. We will see this picture over and over. And the dense contact that it's making with the axon plasma lemma. Lovely. Now this slide just makes the point. See, here you have the axon, here you have the myelin, that's going to come over here, this picture. Here you have the astrocyte in contact with the myelin. Okay, it doesn't look to just be in contact at the node of Ranvier. It likes that spot. It wants to be there. There's going to be a major electrochemical events going on here that are not going on here, but this is going to deal with acidosis and this isn't. And so this is going to have to be in contact with this to create that machine, that neurovascular unit, which supports the neuron. Okay? pH. pH. And then finally, just to speak a word about the microglia. Microglia, the way that I think about microglia is they are the your resident macrophages. So in any tissue, you have to have resident macrophages for a lot of functions, immune functions, epitope presenting functions, type 2 histocompatibility stuff, all right, scavengers, but more complicated than just scavengers, receiving information from cytokines and responding to lots of different stimuli that, we're only beginning to understand with COVID. Is teaching us a lot. So I would think of the microglia as the brain's resident macrophage. Under normal healthy conditions, they don't have a lot to do, and you shouldn't be there. They shouldn't be there. Under conditions of infection, tumor, trauma, they're going to be activated. We're going to need them to clean up the mess that has been created, whether it's an inflammatory mess or blood products or whatever it is. The astrocyte, the glial cell, the neuron are certainly not going to be doing this. And somebody's going to have to come and help. And that's the microglia. So, and much more, of course, sophisticated than just that. But that's for another day. Pictures of microglia rosettes, picture of, digesting undigestibles and things like that. When it gets to the point that the microglia can't digest it as a prion protein, you're going to get in trouble. Then it will never get rid of the prion protein, and that will start to accumulate. And then you're going to have a problem. Okay, microglia, 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 mess, lysosomes, right? Peroxisomes, sure, because they're cleaning up. They're digesting things that nobody else can take care of. All right, so that's it. Now, next week we're going to start looking at macroscopic anatomy. Just 1 second for me. We're gonna start with macroscopic anatomy. We'll look at the cerebrum next week as a unit, and then we'll also get to brainstem as a unit, and then posterior fossa, little embryology, and then we'll start getting to regional anatomy, neuroanatomy. Thank you, everybody. That's it. That about wraps it up.
Thank you.